On behalf of the College of Arts and Sciences at Lawrence Technological University, I'd like to welcome you this evening. And to begin our program, I'd like to turn over the podium to our president, Dr. Verinder Madgil.
Thank you. Thank you, Dean. Good evening, everyone. Are we here? Good evening. You are here now. Thank you very much. I wanted to share my enthusiasm with you. I think your response indicated you are enthused for this evening, which is a delight to have, to listen to, and perhaps a historic one. As you know, that this is one of our premier events at Lawrence Tech. We have had the pleasure and honor of receiving attention and lectures from the preeminent scientists, thought leaders, social leaders, politicians, and presidents of universities, the Nobel Prize winners, and would-be Nobel Prize winners. This simply shows that this particular event or platform is very unique because we bring the very best that we can have access to. And that today evening is such occasion that we have one of the very best and most prominent and person with great credential, protein biochemist, biophysicist, whose work will enthrall you, and you should wait for that. I also want to acknowledge Holly Foundation that has really helped us to install this lecture series. We appreciate that honor. And in the person who was with Doctoral Edison for a long time, as you read on the board, and made tremendous contribution to this area, to every part of society in Detroit area that is touched by his generosity and leadership. So we appreciate him as well. Even though he could not enjoy the lecture, but I know he's watching us. We hope you'll continue to visit Lawrence Tech for this and other events. We are on the swing of a great renaissance in the institution. If you come here every year, you see every year there is something new you see. We have great professors, new buildings, new facilities. We are in public eye a little more because of your generosity and attention, and this lecture adds to that glory. So I thank you, and I will now invite our leader in College of Arts and Sciences, Dr. Shopping Moore, who will introduce the speaker. Dr. Moore.
Thank you, Dr. Madgil. It is with distinct pleasure and honor that I present to you the 2018 lecture of Cisler lecture, Dr. Peter Walter. Peter is a dear friend and colleague of mine for over 30 years. Has it been 30 years? We first met at University of California in San Francisco, where I was a postdoctoral fellow, and Peter was a young assistant professor. And he climbed through the ranks very rapidly, is now a distinguished professor and past chair in the Department of Biochemistry and Biophysics at University of California in San Francisco. Peter is a Howard Hughes Medical Institute investigator and also a member of the National Academy of Sciences. As you know, these are two highest honors one can attain in the sciences. Peter is a German-American. He was born in Berlin, and he attended Free University of Berlin, where he obtained a bachelor's degree in chemistry. He then went to the United States, and he attended Vanderbilt University in Tennessee, where he earned a master in organic chemistry. Now, Peter was not exactly enchanted with Vanderbilt, so he was about to leave Tennessee when he got admitted to Rockefeller University for a PhD program, and that changed his life, I think. So he went to New York and went to work with Dr. Gunther Blobel, who is at Rockefeller University. He studied under him, under his direction for his PhD work. Dr. Gunther Blobel is a leading cell biologist. They decide together to tackle a very important but very challenging question, And that is, they started by asking a very basic question, is how proteins know where to go inside the cell. As you know, there are over 20,000 proteins, different proteins inside the cell. And they each are targeted, they're made in the same organelle called the ribosomes. And then they are targeted to different parts of the cells to exert their function. So, for example, digestive enzymes are targeted to degraded compartment called lysosome. And proteins involved in energy production are targeted to the power plant, mitochondria. So Peter and Gunther decided to work on a set of proteins there to be exported from the cells. And these are proteins that they traverse through a series of compartments and reach the cell surface. And so before Peter started, the lab has established two very important observations. One is that all exported proteins start their journey at what's called endoplasmic reticulum, which is ER, or it's an organelle that's present inside the cell. So this is, in other words, they start the journey from the first station in the pathway, which is called ER, and then they move through a consecutive compartments until they reach the cell surface. The second thing that they discover, which is very important, is that all these exported proteins all contain a signal peptide, which is a string of amino acid that's added onto one end of the protein. And these signal peptides serve as an address signal to enable them to be targeted to the ER membrane. So if you want, this is by analogy, you can see it as a postal system. So the signal peptide is the zip code, whereas the cellular machinery that interprets the signal is the postal service. So what's going on is that at that time, it is not known what a cellular machinery is that interprets this signal. And what Peter did is to answer this question. Now, this is a very, very competitive field at the time. So many labs around the world are working around this. So rumor has it, I don't know if it's true or not, that his advisor, Gunther Blobel, taped a note next to his desk that says, "You are one, and there are many. If you don't work day and night, they will crush you like a roach." Is that true? I think so.
It's a true quote.
So not only did Peter not crushed like a roach, but his profound discovery really opened up a new field in cell biology. So what Peter found was two macromolecular complexes. One is called SRP, and the other is called SRP receptor. SRP stands for signal recognition particle. So SRP receptor is a receptor. So what he showed very elegantly is that proteins that are targeted to the endoplasmic reticulum are recognized by the SRP protein or macromolecular complex that then target them to the receptor on the ER membrane, and that's how they are targeted to their final destination. And this work is so profound that it contributed greatly to his advisors winning the Nobel Prize in 1999 for medicine and physiology or medicine. And a lot of us who know Peter felt that he should have gotten the Nobel Prize for that work, or at least share the Nobel Prize with his advisor. But that does not bother Peter or so he went on and do bigger and better things. So when he went to University of California at San Francisco, he started another set of experiments in research. So again, he asked a very fundamental, basic question, and that is, how do cells know how much organelles to make? Okay, so he was fascinated, I think, by the electron microscope pictures of pancreatic cells. Pancreatic cells make a lot of exported proteins, so they actually have a lot of, an abundance of the endoplasmic reticulum membrane. And he wonders why the cells make so much of the ER membrane. Okay, this is a very fundamental question. You don't know what it's going to lead to, and, but he asked the question anyway. And his hypothesis is that something in the ER membrane sensed that the ER capacity is exceeded, and therefore signaled the cell nucleus to make more of the ER to compensate for this capacity. And it turns out that something is unfolded proteins. So he, at UCSF, he did a series of very elegant and beautiful experiments that show something called the unfolded protein response. And that is how the unfolded proteins somehow translate the signal to the nucleus and to the cytosol to combat this stress response. And he's going to talk about this, and he also has discovered that one of these pathways shared a lot of common characteristics with other stresses inside the cell. So at the protein production level. So he has identified a very key component of this pathway, and it turns out to be a potential therapeutic intervention for many diseases such as cancer, such as Alzheimer, such as diabetes. So he's going to talk about these new discovery. He told me that what his talk is going to be is actually about things that are discovered only within the year. So, and some of them are not published yet. So we're in for a very, very, very amazing treat. So I want to make sure that you know the take-home lesson from Peter's talk, and that is basic research is really, really important. And when he started, every experiment he started with a very basic question, and you don't necessarily know where it's going to lead to until you started getting results and following your nose, and then in the end, you get something that's going to have great impact on humankind. So some of you are at the last year's Cisler lecture. Dr. Mark Schlitzel from University of Michigan talked about curiosity-driven research, and that has the same take-home lesson, and that is you start with something really, really basic. You don't know where it's going to lead you, and in the end, you'll be surprised. So for students in the audience, I want to let you know that if your parents, your friends, and your relatives ask you, why in the world are you studying that? You now have the answer to that, okay? So lastly, I want to say that Peter has received numerous scientific awards and prizes, and I'm going to just cite a few of them. The Wiley Prize in Biomedical Sciences, the Gardner Award, the E.B. Wilson Medal, the Otto Warburg Medal, the Damstetter Prize, Ernst June Prize, Shaw Prize, Breakthrough Prize in Life Sciences, and the Volchek Prize. The last one is to honor someone who is an immigrant who has done tremendous work to influence the U.S., the United States. So also, he is the winner of the 2014 Lasker Award, which is known as the American Nobel. So they pride themselves in identifying the future Nobel Prize winner. And we all know, actually a lot of Peter's colleagues think that he is going to be a future Nobel Prize winner. And with the work that he's going to present to us, you can see why. So without further ado, I would like to invite Peter to the stage, and he will deliver the 2018 Cisler lecture. Thank you.
Thank you so much for this overly generous introduction. Let me just correct one little factoid here. Is when I applied for graduate school at the Rockefeller University, I was rejected. I only slipped in on the waiting list. And that's actually one of the messages of my talk, that science and life aren't linear. The path is meandering and full of surprises. And it's really this what makes me so happy in this profession. It is so exciting to go somewhere where nobody has been before and to see things that nobody has seen before. And it's not a job, it's really a passion. So my students often ask me, well, if all this recognition comes because of discoveries, and discoveries by their very definition are not predictable. Can you turn down the sound a little bit? It sort of echoes. Then our careers entirely depend on dumb luck. And I think this is precisely the wrong way to look at it. There's a much better word in the English language, and that is, if this thing works, that word is serendipity. So who in the audience knows where this word comes from? It's actually a very recent word in the English language. It was introduced in the late 1700s by an English nobleman who wrote a letter to a friend in which he recited an old Far Eastern fairy tale, The Three Princes of Serendip. And as the story goes, there are three princes. The big king has three sons, and he sends the sons into the world to discover the secrets of life. And they walk down this road, and they meet, this is actually not in the Far East, this is in Chile, but take it for what it is. It's a sandy road. And they meet a camel driver, and he says, "I've lost my camel, have you seen it?" And they say, "No, we haven't seen your camel. But is it the camel that's blind on the left eye, that's missing a tooth, that's limping on the right leg, that's carrying butter on the right, that's carrying honey on the left?" And they get promptly thrown in jail. Because if they know so much about this animal, they surely must have stolen it. It turns out, eventually, the camel is found, and they really had never seen it. But they made all these deductions about the image of the beast by following little clues that they had observed on the side of the road. So there were clumps of grass that were half chewed, about the size of a camel tooth. So the camel must have been missing a tooth, and it was just falling out. The grass on one side of the road was growing much higher than on the other, but it was eaten where it wasn't growing so well. So the animal must have been blind on the left eye. There were only 3 footsteps in the sands. There were ants attracted where the butter had dripped. There were bees attracted where the honey had dripped. So they made all these conclusions from little observations that they made as they went along that at the time didn't make any sense. But as they went further and learned about that this animal is missing, they put it all together into a working hypothesis describing the animal, and made this remarkable conclusion. That's precisely what we do every day in the lab. Right? We make observation. Science becomes most exciting when things don't make sense, when we have paradoxes, when our predictions turn out not to be complete, but there are new things to be discovered. So serendipity is not dumb luck, but it's defined as making discoveries by accident and, importantly, cleverness of things you were not looking for. So I think it's a wonderful word for you young scientists out there. Serendipity is our friend in this business. Of course, eventually the princes are rewarded for their cleverness. They get their princesses, and they lived happily ever after. So this story has a good ending. Now let me start out by just putting what gives me awe every time we think about it. Is the miracle of our bodies, or pretty much every multicellular organism, is that the human body contains some 10 to the 13 cells. This is about 1,000 times more than we have people on the planet. These cells are of some 200 different cell types, yet all of these cells, live in mutual supportive symbiosis. And this is really a remarkable thing. I mean, if you think about it, 1,000 times more than we have people on the planet. And if one of the cells in our bodies turns selfish, then we have cancer. So it is an absolutely remarkable fact that we don't all have cancer, right? That the body has these corrective mechanisms by which to prevent that. What's equally remarkable is that every organism is developed from a single egg cell. This egg cell gets fertilized. And then it develops into the organism that's specified by the genome, into a mouse, or into this algae, or into this, I think it's a sea urchin thing here. So all the information of making all the different cell types in the fully formed organisms is encoded in the egg. And I put a movie in here just to show you how really remarkable this phenomenon is. There's a fertilized frog egg, or 3 of them. And you see that as they start dividing, they divide in perfect synchrony, so program is carried out. They become this larger cell mass, confined by the boundaries of the eggshell. And then there are remarkable rearrangements that happen, again, synchronously in development, where cells now become different from one another, and cells have to know what their neighbors are, where they are in the organism. You now see organs develop, the eye spot, and then eventually they differentiate into forming an intact tadpole. It gets getting too tight in here, so eventually they figure out a way of getting out of there. And this is a process that takes a few days, and there is a little tadpole born. Isn't that amazing? And this is what we want to understand. All of the cells in our body, by first approximation, have the same DNA, the same genome. Yet all the different cells have to be vastly different from one another. And there are then external and internal signals that determine which cell is going to become what. And they have to listen to one another and talk to one another to determine the correct cell structure, the correct cell behavior, the correct cell fate. So different cell fates are programmed. Some cells are programmed to die. The reason why we don't have webs between our fingers is because the cells that formed this connective tissue were programmed to die and therefore allowed our digits to develop separately. And all these different functions that make cells different from one another are carried out by proteins. And as Xiaoping already alluded to, proteins are really the machinery of the cells. They're not just food, as you see in most of the advertisement, but the reason why we eat proteins is to break them down into amino acids so we can build our own proteins that then make the cells function. So they're machines that make cells work. They are the enzymes that convert metabolites. They are the motor proteins that create mechanical forces. They function as structural proteins to provide the rigidity, mechanical rigidity to cells. And they function as receptors and signaling molecules that interpret the information, that transmit the information between different cells, so they know where they are in the body and what they have to do with this information. And then we have gene regulatory proteins that switch on and off a particular set of genes in the appropriate cells, so one cell can become different from another, and that they can respond properly to different environmental stimuli. And this list could be continued. So the basic idea is proteins are really the basic building blocks, the machines that make all of this happen. And each of our cells, on average, makes about 1.5 million new proteins every hour. So the task of making these proteins, making sure that they are correctly assembled, is really a massive undertaking. So proteins are made, encoded by the genome. The genome is transcribed into a messenger RNA that then contains the information of one gene at a time. And this information is translated, interpreted by the ribosome, which takes the genomic information and translates it into a series of amino acids, the building block of proteins, that get assembled in a particular order. So there are 20 of these different building blocks, and they get arranged in this linear sequence, which is emerging from the ribosome and is specified by the mRNA that interprets it. Now, once this chain has been completed and released from the ribosome, if we stretch it out, it's about this side, and then has to fold up. And this is drawn to scale. So a protein chain of this length compacts itself to the folded protein that eventually ends up in the cell. And so there are some regions here that are helical, some regions that are contacting each other as individual strands. And the reason why this folding up is important is that now certain amino acids can come together in space and form these regions that we call active sites. And these are the active sites with which proteins fulfill their functions. That's where metabolites bind and get transformed. This was a view many years ago that this would be a linear sequence. And in fact, a Nobel Prize was given to the fact that the amino acid sequence itself specifies the folded structure of a protein. And for simple proteins, that's true. We can just fold them up. But in truth, there are many intermediates, and it is a much more complicated landscape of protein assembly and folding, in that there are folding intermediates that form initially. There's not a stretched out chain, but certain elements, like these helices, form first, and then they fold into the final structure. The final structure is not static, but these are really breathing entities that can unfold again into these intermediates. Proteins don't live forever. They get degraded into bits and pieces with different half-times, so different proteins have different lives. And from all of these stages, they can assume misfolded states that then either can be remodeled or can be degraded. So it's a very complicated landscape. All these intermediates can start aggregating either into amorphous structures, non-specifically. Just think of an egg white, right? A boiled egg. The egg whites come together here, forming these structures. Or they can form ordered aggregates, such as amyloids, which as you know, are many of the prion diseases and neurodegenerative diseases, where the accumulation of these structures leads to the destruction of nerve cells. And some of these structures are more resistant to degradation than others, so they can be degraded and finally be recycled again. This is a very active field currently of investigation. As a matter of fact, over the 5 years, there have been 5 breakthrough prizes awarded to various aspects of this field, so it's really an ongoing active research. And there are about 1,000 enzymes or so in every one of our cells that are dedicated to this process of protein quality control. So they mediate these various interactions and various conformational changes that the proteins undergo. So there are chaperones, for example, that prevent the aggregation by binding to misfolded protein structures and preventing them from doing nasty things to the cell. And many of these quality control mechanisms that are operating diminish as we age, or they break down in disease. So what I would like to do in this lecture here is give you an idea that this is a largely untapped, enormous opportunity to work on the quality of our proteome and therefore on the health, on the basic health of every one of our cells, so they don't end up in these diseased states, which in this case is actually not a disease, but a condition that affects all of us eventually. So as Xiaoping already alluded to, our work starts in the endoplasmic reticulum, which is a compartment inside the cell in which proteins fold and eventually enter the secretory pathways or other compartments to end up in the cell surface or be secreted from cells. And these are precisely the proteins, right, with which cells communicate with one another. They're signaling molecules that passage like hormones between different cells, or they're the receptors that sit in the plasma membrane and interpret the information in the body, what the cell is going to be programmed to do. So if these proteins misfunction, then you potentially create rogue cells for the body, right, that could do all sorts of damage and behave inappropriately. And probably the best example is cancerous growth. Accordingly then, this compartment in which all of this folding of these proteins has to take place is exquisitely well controlled in cells. And this shows a developmental step here in which this immune cell, a precursor cell, that eventually becomes stimulated to become a plasma cell, so a circulating cell in our bloodstream that makes antibodies to defend ourselves from invading organisms. You see that this event, as the cell prepares to make a lot of proteins that are being secreted, goes hand in hand with this vast amplification of the endoplasmic reticulum. So that in the end, this organelle basically fills the cell wall to wall. And this is just a reflection that this has now become a professional secretory cell that needs to fold a lot of protein. This cell makes its own weight in protein every day, and they all have to pass to the endoplasmic reticulum, where they have to be assembled and folded properly. So this developmental stage then goes hand in hand with the need of the cell. It's estimated that some of these complicated signaling machines that assemble here, that the vast majority of protein chains that are made in the endoplasmic reticulum actually never make it to the cell surface, but rather enter degradative pathways, because the cell puts so much quality control on them that they're considered not to be folded sufficiently correctly. There's an enormous waste of energy, or enormous expenditure of energy, giving you the immediate indication, right, that fidelity of these proteins is far more important than conserving energy. And we've learned now how this step of amplifying the endoplasmic reticulum is controlled, and that's work we've been doing for the last 20 years or so. And this is this pathway called the unfolded protein response network, which basically adjusts the amount of endoplasmic reticulum that the cell has to fold its proteins. So if we enlarge this portion of the endoplasmic reticulum here, there are three principal pathways. Each of these pathways is made up by proteins that act as sensors that have to figure out, that sit in the membrane, span the membrane of this compartment, and figure out on one side what's going on in the lumen of the endoplasmic reticulum. Do we have enough protein folding capacity, or is the load imbalanced? For at least two of these different pathways here, we've learned that the unfolded proteins themselves are ligands that activate these receptors and activate signaling pathways downstream. So when you don't have enough protein folding capacity, you accumulate these unfolded or misfolded proteins, and they then directly activate these receptors downstream. And then in pathways that have been occupying us for the last 20 years or so, the information is then passed downstream by very interesting mechanisms to activate these gene regulatory proteins that then, in response to conditions inside the endoplasmic reticulum, activate a particular set of genes. What has been so amazing is that each of these black boxes, which I'm not going to go into the detail for this talk, contain the most amazing mechanisms that are fascinating for the aficionados in the field. So this IRE1 here signals via a mechanism of non-conventional mRNA splicing. That's my daughter, by the way. The PERK pathway here functions through a mechanism of translational control. And finally, ATF6 here functions through a mechanism of regulated proteolysis, each of which have elements that are entirely new in biology and that came out of deciphering this path of discovery of just putting one foot in front of the other. And then these gene regulatory proteins, they collaborate with one another to upregulate an appropriate set of target genes in the genome to make more endoplasmic reticulum. So we end up with an imbalance. We don't have enough protein folding capacity in the endoplasmic reticulum. The load is higher than the capacity to deal with these proteins. So we now balance the load and capacity by making more endoplasmic reticulum, making more chaperones, degrading these things, making fewer proteins that then increase the load on these compartments. So basically, we are bringing the system back into homeostasis. If this cannot be achieved, however, then the cell makes a very exciting decision of engaging in a suicide program, programmed cell death. So rather than making a cell that is potentially a rogue cell because it displays misfolded proteins on the surface, the entire cell is programmed to remove itself from the organism. Again, an exciting molecular question, right? How does a cell decide? Can I fix the problem or should I kill myself? And we've learned a lot about this process in recent years, where there are timers built into these pathways that allow the cell to first try to fix the problem, and then if in a certain time window this cannot be achieved, then the apoptotic, the cell death programs are induced. This is an example here of cells in which the unfolded protein response is induced. The cells cannot handle it, and you see that these cells here grow nicely. They form these confluent layers, whereas these cells just disintegrate and become these cell debris that's floating around afterwards. So this is just sort of awful to watch, right? How these beautiful cells die. So this is the cell death. We've learned a lot about the process. And this just shows you a little, it's a coarse-grained simulation of the process of one of the pathways that we've studied most in the lab. So that's the IRE1 pathway. And you see the membrane protein sits here in the membrane. These are members from my lab. And if everything is fine, everything is calm in the endoplasmic reticulum, you see nothing much is going on. The receptors are silent. And then unfolded proteins accumulate. There's too much load. And now you see that these sensor molecules become very agitated. They have this little capacity here of putting phosphate groups on each other. So see that they transphosphorylate. And now they're active. Now along comes in the mRNA for the resolution to the problem that encodes this transcription factor, but it has this intron in it that needs to be carved out. Intron floats away. Here comes another enzyme that puts the ends that are generated back together. That's a tRNA ligase. And now you've created this non-spliced mRNA that enters the ribosome, gets translated. You see IRE1 calms down again. They make proteins. Phosphate is lost. And at the same time, now proteins start entering these vesicles here that bud from the endoplasmic reticulum. So this is what we do every day. So to summarize, then the unfolded protein response in health we think of it as a finely tuned governor of protein health. Whereas in disease, this balance between life and death can be altered. And it can err on either side. In some cases, it's a rogue cell executor that kills cells that we would rather have stay alive. And maybe the most intuitive example are protein folding diseases, where patients contain a mutation in proteins that enter the endoplasmic reticulum, such as retinitis pigmentosa, where the light receptor in your retina is encoded with a mutation. These proteins are made in a certain step in eye development. They enter the endoplasmic reticulum. The cell cannot fix the problem because it's a mutation. So not only do you have a protein that's not quite functional, but the cell, because it cannot fix it, gets frustrated with the accumulation of unfolded protein and kills itself. So the entire retina degenerates in this inherited form of blindness. We have adult onset diabetes, type 2 diabetes. We are asking the beta cells of our pancreas to make ever increasing amount of insulin, eventually exceeding their secretory capacity, their folding capacity. The cell dies of programmed cell death. And of course, when one cell dies, the cell next door is asked to make even more insulin, so it becomes a spiral downwards. But the unfolded protein response can also err on the side of a rogue cell protector that keeps cells alive that we would rather have die. And examples of that are virus infections, where many viruses use this amplification of the endoplasmic reticulum, so they have enough capacity to make membrane proteins to engulf them, to encode themselves. And so if we could develop an agent that could just subtly shift the balance between life and death, we could potentially make a very broad spectrum antiviral from which the virus could never mutate away from, because it's a cellular function that is abusing here for its own good. And then finally, we have cancer, where cancer cells evolve in our bodies. They form tumors. They forget how to interpret information properly. But since the evolution happens, the induction of oncogenes, the loss of tumor suppressor genes, which causes cancer, this evolution is not a directed process, right? It's a process that happens by random changes in the genome. Cancer cells become genomically unstable as they become more and more aggressive. And these evolutionary changes, because they're not directed, not only change proteins that are then good for the cancer or help it to grow, but they also create a lot of so-called passenger mutations. So in other words, other proteins that are not folded properly, that are turning on these unfolded protein response pathways and turning on heat shock and other stress pathways in the cell. So a property of every cancer cell, right, is to have a very unstable and a very defective proteome that depends on these stress pathways to survive, to still function properly. So cancers then derive a growth advantage out of these stress pathways that allow them to deal with this large load of unfolded proteins that they have in various compartments. So I want to show you this amazing result from a collaboration that we have. So basically, our approach has been to learn as much as possible mechanistically about these different pathways and see if we can shift this balance, life-death, depending on the disease condition that we're having. And in this case, we're looking at a cancer model with our collaborators at Genentech. So we've moved this now from academia into biotechnology, in which we look at a cancer, multiple myeloma, which is a cancer of these very plasma cells that I've shown you before that secrete the antibody molecules. It's a very aggressive blood cancer. And in this case, these cancer cells, human cancer cells, are injected into this mouse. A tumor is established. And then if you look a few days later, you see that this cancer very aggressively spreads to the mouse. In this case, the cells are labeled with a light-producing protein, so we can actually follow them in the live animal. And you see that this signal vastly amplifies, and the metastases go everywhere into the brain and various parts of the body. And in this time window that has been looked at here, some of the cancers here have grown, the tumor mass has grown 20-fold, 5 to 20-fold in these animals. So we do the same in another mouse, make sure the tumor is established, and then we inject it the mouse with an inhibitor to this IRA1 pathway that I just showed you the movie of, that my lab acted out there. And this is the amazing result. The tumors, not only does the tumor not grow, but it actually is eliminated from the animal. So I think this is very early days, but this is very promising. And we are actively developing that. We screened for other molecules that affect other branches of the unfolded protein response, just to see what we can do. And from the PERK pathway, so that was the middle pathway of the cartoon I showed you, we isolated this marvelous molecule here, which we call ISRIP, for integrated stress response inhibitor. And I think this is truly beautiful, this molecule. Right? I trained as a chemist initially, so for me this is really amazing. So we went through some 100,000 molecules, different chemicals, and we isolated this single one that shuts down the PERK pathway, signaling through this PERK pathway of the unfolded protein response. There's beauty in twofold symmetry, right? It's a twofold symmetric molecule, so these two arms are identical, joined by this cyclohexane ring. And because we can substitute this ring in two different isoforms here, both in cis and in trans, the cis form is entirely inactive, the trans form has all the activity, so we have virtually identical chemical matter as wonderful controls in these experiments. So what does this thing do to shut down this pathway? Well, it told us, the molecule already told us that it's twofold symmetric. And whenever we changed something here and we made the same change here, it either made it better in both cases or it made it worse in both cases. So this molecule really indicated to us, I want to be twofold symmetric. And we've now solved the structure, which is going to come out in 2 weeks in a nice paper, of this drug molecule bound to its target in the cell. And this is done in collaboration with Adam Frost and my graduate students at UCSF. And there in the middle of this huge protein complex, we see this little drug bound. This is cryo-electron microscopy. So we can literally see these days protein complexes in the microscope and average the things. And what you see here, maybe you can see it, but this large protein complex, it actually contains 10 different subunits. It's bound together via this twofold symmetric interface. So this part and this part are identical. And our little ISRIP molecule here acts as a clamp to bring these two pentameric complexes together. So it's a molecular clamp, a molecular staple. And when it does that, it makes this component that has become limiting under conditions of activating the PERK pathway, it makes this complex sufficiently more active that it now overcomes this inhibition. So we have a molecule here that inhibits an inhibition, right? Which means it's not going to do anything unless this inhibition is on in the first place, which is a very important thing. It's not an inhibitor in this sense, but it's an activator of this enzyme, and it inhibits the inhibition. So the surprise that came out of that is it is not toxic. So even so, we are affecting a basic pathway that's present in every one of our cells. Giving this molecule to an animal shows no, to date we have seen no toxic effects of this molecule. So that's a big surprise. We've also shown in the paper that it's now going to come out in a few weeks, that this molecule is very effective, just like the IRA1 inhibitor, in this case against metastatic prostate cancer. So stay tuned on that one. But the biggest surprise came out of just talking to our colleagues. And in this case, it was a phone call I had with my dear colleague, dear friend, Nahum Sonnenberg at McGill, And in Nahum's lab, Mauro Costamaggioli, who at the time was a postdoc with Nahum and now has his own lab, they discovered that this very pathway that's affected by the drug is involved in the process of memory formation, of cognition, of memory consolidation. So to take short-term memory and to move it into a long-term storage. So we sent them some stuff, and they showed indeed that ISRIP can function as a cognitive enhancer. That's entirely unanticipated. So this is true serendipity here, right? We put no intellectual effort. I had no idea I would ever have to turn into a neurobiologist. And so basically, when it's injected, it allows normal mice to form better long-term memories, which is amazing. So the data look something like that. What we do is we take a mouse and we train it to find a hidden platform in this water maze. This is somewhat milky water, so it cannot see these things. And the mouse orients itself by visual clues around the room and learns how to find this hidden platform, and we just measure the time it takes for it to find the platform. And these are control animals that have been trained over multiple days. It's a light learning regime, so that they don't optimize their performance, so they can actually improve. And you see that they plateau at a certain time here of best performance. And if we inject the drug into these animals, you see that they take much, much shorter. They go straight to the goal. We can show the same in many different other behavioral assays. So this has profound implications. So first of all, I mean, we are improving here the performance of a normal animal. So evolution in this animal has not optimized the process of learning, long-term memory formation. And we can come back to that in the discussion, but it's probably due to the fact that you don't want to remember everything. You don't want to load up your memory with useless detail. The second thing it tells us that there must be a natural break on this memory consolidation in this pathway somehow, and our little drug here can release this break. So we actually have a pharmaceutical agent now which we can enhance, and the question is, can we exploit this increase in cognitive capacity in some diseases? Right? What we don't want to happen is for parents to give it to their children so they perform a little bit better the next day in the exam, right? And I think it's a wonderful example here, something I've never experienced before, is that how quickly one can slither from clean, curiosity-driven research, where we want to understand the molecular mechanism of something, into issues that are quite ethically charged. So for all of you, really take the ethics of what you're doing seriously. It can have profound consequences. ISRIP, the taste kids love. Could we please have some more? No. But there are many real clinical implications, and we may open new paths here to explore how memories are stored and lost, which today nobody can really say how this is accomplished. And we can explore potential therapies in disease with cognitive defects. And these cognitive defects, right? I mean, all of you are familiar with neurodegenerative diseases, Alzheimer's, Parkinson's, and so on and so forth. The hope here will not be to cure these diseases, but maybe to make the life quality of these patients better for a longer period. We have brain injuries, mechanical brain injuries. And finally, as is increasingly becoming apparent to me, its cognitive decline is also a function of normal aging. So I hope whatever we do here happens fast enough. So I want to show you, towards the end here, experiments that we've done together with Susanna Rossi. She works at the Trauma Center at UCSF. And she has a mouse model of traumatic brain injuries. And basically what we are doing here is we are training mice, again, in a water maze. Again, the mouse orients itself by visual clues around the room, and this is a control mouse here that has learned how to find the hidden platform. You see it's very quick. And this mouse has been subjected to traumatic brain injury. There's a mechanical injury to the brain, and it basically has no idea where it's going. It's completely incapable of learning anything. And I can show, this movie runs for quite a while here. Eventually we rescue the mouse, so no mouse was lost in these experiments. So in this case, the water maze has these different arms. And what we can do is we simply measure how often the mouse swims down the wrong arm, so the number of errors it makes. And in this case, the experiment is done here that the mice, the injury is inflicted, and then the mice are given 4 weeks to recover. So at that point, all inflammation has declined. They run around with the same speed. They swim with the same speed. They balance well. So you couldn't tell anything, but they cannot learn anything. They're basically, all their long-term memory is gone. So this is so they make on average here 3 to 5 mistakes, and the controls in this particular time point here made 1. So if after 1 month, we now inject the drug into these mice, the effects are entirely reversed. So they learn now as indistinguishable, statistically indistinguishable from the uninjured counterparts. You see that even the controls learn better, but this is a really, really impacting results here. So ISRIP can correct cognitive defects after brain injuries, even if given a long term after the injury. So I think what this tells us is that what the injury did here is it turned on these responses, but it's a maladaptive way in which it's doing. It's trying to fix the problem, but in fixing the problem, it induced new problems. And by now killing the response, in this case, we can affect the outcome of the disease. This was too good to be true, so we didn't believe the results. So we wanted to make it a little bit more difficult to the mouse. So we used this second behavioral test here. And basically what you do is you have this big table and it has 40 holes in it. And only one of the holes has a little escape hatch underneath where the mouse can hide and be cozy. And then you shine bright light and you make some noise, and the mouse wants to hide somewhere. And it finds this, learns where to find this escape hatch, and is happy down there. If you do the same with animals that have received the brain injury, you can see that they walk a random path. They basically haven't learned how to do that. So you do this assay, and then the next day you repeat it, but now you change the position of the escape hatch. So now the mouse has to forget what it learned the day before. It has to relearn, but at the same time, it also has learned how to learn. So in other words, it spends less and less time exploring. It is much more quick at accomplishing this task. So it's like you finding your car in the parking garage. Right? The parking garage is always the same, but your car is in a different position. And admittedly, I look more like this. And the results are, again, very clear-cut. ISRIP reverses the effects of the traumatic brain injury when given a long time after the injury has occurred. So what do we do with this? Well, it's fantastic news for all the mice. Mouse brains are very different from human brains, so we have no idea whether any of this will translate into human biology. But I think we are very hopeful, and we've licensed it now to a biotech company down the peninsula who are developing it for preclinical studies. But in the meantime, there are other ways you can protect your football players from unnecessary harm. Okay, so let me close with the main message I wanted to leave you here. is that our strategy has been to understand the precise molecular mechanism that control the fates of cell proteins, to modulate these mechanisms in as surgically as possible with our small molecules that we spend a lot of effort, and we are luckily funded by the Howard Hughes Institute to take this risky approach in various disease models. And then hopefully some of these results will translate from animal models to human and then lead to, after assessment of safety and utility for therapeutic intervention, to clinical trials in which we can check them out. This is sort of a smooth transition, I would say, between academia and biotech, where the risky stuff we can do much better in academia. Students are wonderful at going down these roads, whereas down here you always get the bean counters who need to see the profits of the company at the end. So these approaches that we do in academia open the doors and then de-risk them as we go along before they're eventually picked up by biotech companies. Our 10-year dreams is that we develop novel cures for cancers, cognitive disorders, and other diseases by targeting these Achilles heels of entire disease classes. But you may have noticed that this is entirely the opposite as precision medicine is doing for cancer. We are not subdividing the cancers, but we are focusing in on an aspect that's common to all cancers. All cancers have to evolve to become more aggressive and to become invasive. And this actually, it's similar to what's happening in cancer immunotherapy, where these defects that cancer cells display are recognized by the immune system as defective, and therefore cancers are targeted. But we're looking at the inside of the cell, right? We're looking at the endogenous mechanisms that every cell already has to deal with these things. And then we hope that we can develop preventive measures to keep cell proteins healthy, the proteome of the cells in a healthy state, and nudge society from the current state of healthcare, which really is sick care, right? You go to the doctor when you're sick. You don't go to the doctor when you're healthy, to an actual healthcare that if we take care of our proteins, we're going to live a happier life. And since these pathways are being shut down as we age, I think we really have an opportunity to affect these aging programs. How can we improve our chances for success in this business? Well, this is a different game than one I grew up with. It's called soccer, where the goal is to take this ball and to kick it into this goal. I'm not sure that explanation in America. In Europe, I don't need to say that. And how to increase your chances for success? One is to increase the precision of the process, and that's, I think, what biotech is doing fantastically right now, right? They're developing really sharp boundaries to the therapies, to the drug development. But I think there are other ways we can do that. We could just put more balls into play, right? Like attacking the different branches of the unfolded protein response all simultaneously and see what happens. Certainly your chances of hitting the goal with one of those is increased. And then the other idea that we are pursuing in our approaches, you just put up more goals. In other words, I showed you, right, that targeting the same molecular pathways here can affect both cognition, neurodegenerative diseases, and cancer alike. And it can be extended to the other diseases I mentioned before. So as Xiaoping alluded to, since I'm German, I have to use at least one German word here. So Schiller used this edle Wissensbegierde as a noble quest for pure knowledge. We set out doing all that out of curiosity-driven research. We had no idea that this would connect fruitfully to any disease setting. Our approaches initially were entirely disease agnostic. We want to understand how these molecules function, how the signals are being transduced, how we can interfere. They were not predictably translational. That's very important because every funding agency wants to see the translatability of what you're proposing today, which is, in my view, entirely wrong and entirely misguided. We could not have predicted this, so we would have never been funded had it not been for some mechanisms. And it's an endless task. There is every question we've answered, we found many more questions that now become exciting and unanswered. So the philosophy in the lab is this one. This is my role here. Never mind what's holding it up, keep hammering. Over here you see the students and postdocs hammering away. And my role is not only to provide the material and the funding to do all this research, but the most important part is actually to instill confidence that eventually we'll reach terra firma again over on this side. That brings me to the last thing here, a quote from Einstein: as the circle of light increases, so does the circumference of darkness around it. And it's this circumference of darkness that's ever increasing, right, in which all the young people in the audience, that's where you should seek the challenging questions. It's not in the lightness, it's not what we already know. It's not the results you're expecting. It's the paradoxes you see on the side of the road and try to put them together in some form or another that gets you further. Typical day in the lab. You're given these beautiful equipments. You go and dig. You dig, you dig, you dig. Get some good signals. The signal can be, right, your advisor telling you that you should keep digging, or otherwise you will be crushed like a roach. Typical Sunday afternoon. And then sometimes doubt sets in. Right? And this is what you should not do. You should not ignore these hints and the advice you've been getting, because there's so much to be discovered. So I'll leave you with keep digging and give serendipity a chance. Thanks to the lab. These are the people who have done the work. And I think I alluded to it already, but this has what I told you today, what has accumulated over the last few years, has been a 25-year journey. And these are all the people who, in some way or another, contributed to it. So I did nothing for this. These are the credit goes to all of them. Thank you so much. I'm happy to answer questions.
Yeah, we have a little time for questions. We have two microphones here, Tracy here, and Julie. Where was Julie hiding? In the back. So you just raise your hand and we'll get microphones to you.
Thank you for the great talk. In the movie with the mice, you said that some of them had traumatic brain injury. Now, since this was a discovery about memory, does that mean that the injury was inflicted on some particular cells in the hippocampus?
The injury was inflicted frontally, and the cognitive defects spread, but it's hippocampal learning that we're observing here. So it's a general inability to learn. In this case, we measured spatial learning. We've also done it for fear conditioning, both auditory, which is processed in the amygdala, or contextual fear conditioning. So different parts of the brain are all affected by that.
Okay, thank you.
It's probably a general consequence of brain inflammation that, in this case, has become maladaptive to the...
Hi, that was a very nice talk. I had a question regarding the...
Where are you?
I'm way back here.
Okay.
Regarding the cognitive improvements, and because you mentioned like Parkinson's and all those kinds of diseases, I wonder if the molecule that you are targeting, which you're focused on, do you think, because it's my understanding that copper, right, plays an important role in the symptoms for Parkinson's. Do you think that the molecule that you have is actually serving as some sort of chelator, you know, binding to copper, possibly?
Yeah. No, we're pretty sure that what you're seeing here is the target of the molecule that has to engage with this particular target. Because in every case we've looked, we have both genetic evidence and pharmacological evidence that points in the very same direction. So we are sure it relates to the amount of the activation of the pathway downstream of this PERK unfolded protein sensor. So we think that at least for the cognition that we're monitoring here, the effect is pretty direct. It gives us great confidence that the drug actually acts on target. It's actually a very, very powerful molecule. What we don't know currently is whether the effects we are seeing are actually in the neurons. We have evidence for that, but could also be in glial cells or astrocytes or infiltrating macrophages, because the drug is given systemically. So what we're doing right now is we are building mosaic mice which have drug-resistant target proteins expressed in certain cell types. So we can sort this out, distinguish this cleanly, whether these are really synaptic strengthening under these conditions.
Hello? Okay. Is the effect temporary or?
Yeah, it's a very, very good question. So I tell you one experiment that has been done, I think, twice now, but it's really preliminary. And that's where we've improved the cognition on aged animals, the equivalent of, I think, 80-year-old human. We've trained them in one task while we were injecting the drug, and then assayed memory 10 days later. And they performed significantly better. We then let them sit for 3 weeks. No more drug injection. Trained them in a different task, and they learned significantly better. Oh, okay, so it may be that the hope is that the most positive interpretation, that like this bridge, right? I'm giving you my optimistic spiel here, is that that there is a pathway that's maladaptive that's being turned on, and basically the action of the drug is like pushing a reset button. Yeah. That it brings the system back into a more homeostatic mode, and as such, then cognition is improved. We'll see if this holds up. But the effects I showed you for the water maze and the traumatic brain injury here, they last for at least 2 weeks after the injection. No more. The memories that are formed last.
Oh, thank you. Thank you.
Dr. Walter, very thought-provoking lecture, so thank you very much. I think it excites everyone in the audience to explore, and particularly our students. My question is that would you consider, or have you thought about looking at the precocious aging model, progeria, where young people have old people-like symptoms? Yeah. And I think this will be an exciting observation to make, whether some of that can be reversed.
Yeah, yeah. So we have not. What are you doing next week?
I'm with you. Thank you.
So, not yet. There's so much to do, right? I mean, on this particular project, I have 3 people in my lab working on it. So a lot of it is done in collaboration with people who have good models and who we have fun with working with.
Thank you, Doctor, for a very, very interesting presentation. My question has to do with the ISRIS molecule. Did you discover this? Is it an invention? Can it be patented? How will you handle this this thing?
Yeah. So so this molecule came. We wanted to find inhibitors for the PERK pathway. We constructed reporter cells that can, when the pathway is activated, now express luciferase, so are distinguishable from unactivated cells, and we screened some 100,000 small molecules in a library, from a library, for effects of turning this pathway off. That yielded this one molecule. It took about 3 years, so it's a remarkable postdoctoral fellow who did this work. Since the library is a public library, the composition of matter is not patentable. That was available to everybody before. But we have the method of use, and I think the company we licensed our technology to now has worked out different chemistry that makes it a better pharmacological agent, and then will become patentable in that respect. But we have a lot of method of use applications.
We've all heard about the possible relationship of amyloid to a degenerative brain disease. How does your inhibitor of the PERT pathway relate perhaps to amyloid?
So presently we don't know. We've just started a collaboration with Stan Prusner's lab to look at the prion formation in various prion diseases. There's one report I heard that in Alzheimer's it prevents the beta 41 activated Alzheimer's conditions. The question is still in the field whether the amyloid formation itself is the disease-causing agent, or if it's these smaller oligomers that form on their way to becoming amyloid. So the amyloids may actually be a protective way of storing away potential toxic proteins that may just bind up chaperones. And so the short answer is we don't know. It needs to be tested more thoroughly.
Just take a couple more questions. Let's got a couple students on this side, and then we'll come back.
So I had a question about some of the tools and technologies that you use to identify these chemicals that can treat such these diseases. And I was wondering, from your experiences in your lab, what are some of the upcoming technologies that might be able to improve the identification and maturity for finding these chemicals?
Patience. I'm not sure it's an upcoming technology, but as I said, it took some time. And with this particular molecule, it has a story behind it, that once we finally found it, our screening facility decided to throw it out. They triaged against it because it wasn't soluble enough in their mind to ever amount to anything. So it was strictly due to the initiative of my postdoc Kamala Sadrowski, that she decided to ignore the advice of the expert and to fish it back out of the garbage can and say, I want to study it anyway. The resolution to why they wanted to throw it out is that we ended up with this molecule has an in-cell efficacy of 5 nanomolar right out of the screen, which they've never heard of, right? Usually you're happy when you end up with 3 orders of magnitude lower affinity in your initial screens, and then you have to do a lot of medchem to improve the affinity. But this thing came out with 5 nanomolar efficacy right away, so you don't need much in solution to show that it works quite well. We get nice. I think it's becoming easier to do because the screening facilities are in various campuses. You definitely want to do it with some expert at the beginning who can tell you the ins and outs of the robotics that are required. Screening through 100,000 molecules is not trivial. Some libraries are better than others. So if you do it with a Harvard library, many molecules that you identify aren't actually in the library. That's not necessarily what's in the tube. And this needs to all be, if you cannot repurchase the chemical, right, you have to resynthesize it if you have a good hit. So it's a long-term commitment. And we were lucky that Howard Hughes Medical Institute funded this work. I think it has paid off in spades for them. Sorry that I know this was unsatisfying for you, but no magical tricks.
Dr. Walter, thank you for sharing this information. I work in healthcare, and it seems that the drug companies are all about treating symptoms, so it's refreshing to hear an approach such as yours that's focused on a cure. However, I would ask this: what side effects have you witnessed in your research with ISRIP?
With ISRIP, none.
None?
It's magical. I have no excuse, right? This is not what we expected.
Serendipity, right?
Serendipity, yeah.
What company?
The compound, as you saw it here, that's the original compound. It's not quite as soluble as you wanted it for drugs, so it will never go into humans, I don't think. And when we inject it, the vehicle becomes more toxic than the drug.
Yes, sir. This is potentially a game changer. Last question. What company did you license the technology to?
This was licensed to Calico, which is a Google-founded company down the peninsula from San Francisco. It's run by Art Levinston, who used to be the CEO of Genentech. And since Google is funding it, they have very deep pockets and are willing to take risky approaches. This worked incredibly fast, right, for this game, to find somebody in industry who's interested in something that we've only been working with for a few years. So the first paper on this was in 2013.
We're going to have some time afterwards. There's a dessert reception out front. So let's take a moment to thank
I will take the stairs. Yeah. Thank you so much.